Okay, folks, I'm going to tell you a little bit more about the CRISPR-Cas9 story. We're going to go in some more detail about the MDX experiments. Hopefully, this will also jog your memory. And today, when you're doing your 3x3, I want you to think a little bit differently. I'm not going to be asking you specific details about the experiment, but I want you to be learning about what sorts of things they do and why they do them. That's one thing that you should take away from today. And then secondly, you should also be looking at it in terms of how are we following up from previous lectures on the content of this lecture. And that's another thing that you might want to try to do as your 3x3, as well as trying to pick out some other slides. Now, I'm going to leave this up here, and I'm going to post it on your announcements page. But I decided yesterday, taking a look at the class, and I'm pretty happy with the class. You know, I'm hoping that you're in a pretty good place at this part of the semester, so I don't want you to be in a bad place at this later on in the semester. So I took a look at the material, and I started fleshing out the midterm as well as elements of the final exam. And I decided to make it pretty straightforward. The multiple choice, again, I'm going to try to give you some ideas about the style of my multiple choice test questions. But the short answers, I decided this year to make it really easy to understand, where you're going to be not so many definitions, to be quite honest, but more about drawing, labeling vectors and diagrams, as long as you understand them, and then being able to describe things as well as to design an experiment, which many of you know I really like doing. It's more open-ended. I'm very critical of the types of experiments that you design, but ultimately that's something that I hope is good for you down the road. And again, I want the final exam to be even more straightforward than the midterm, and we'll go through that in some more detail as we get a little closer toward the actual midterm. So in many ways, this is, yeah, do you have a question? Yes. Sorry, what percentage of the... So in terms of how many marks in total are going to be assigned, and then right now I think the multiple choice will be roughly half. Yeah? Is that okay for everyone? Roughly half-ish? So let me take you through some of the data that we've looked at before. So this is sort of the third groups of papers that I introduced to you very briefly. I talked to you about CRISPR-Cas9 in the context of Roger Nichols' paper, as well as Fang Zhang's paper. And this is sort of the last group. And this group was an exciting group of papers that came out. 3 separate labs basically coming up with the same idea, basically coming up with exactly the same results. One paper is a little bit different. We won't worry about all the details. And you're going to see these types of figures a lot. In this particular instance, dystrophin, which is sort of the edges that you see here. And here's an example. The edge that you see here is dystrophin in muscle tissue. And you can see in a normal animal, this is what dystrophin should look like in the muscle fibers. And these are degenerating muscle fibers that you see here. Very, very little green staining that you see between the two. This is after you have rescued the dystrophin back using a technique that we'll be talking about today. So in many ways, this is really about threes. So 3 different papers, 3 different individuals, 3 different types of things that come together in this one sort of paper. So one of the things I really want you to take away from today's lecture, we're not going to go through in a lot of detail. To be honest, I asked a few of you just before class started how much molecular backgrounds you remember. I don't need you to go through all of the details about specific sequences that are involved in alternate splice regions, but I do need you to remember what exon skipping is just in general terms. And I'll go through that really briefly with you. And if you're unsure or you can't recall, you can come and talk to me, but it's not going to be an emphasis on the midterm nor on the final exam. It's more about what I cover in lecture that you should be worried about. I'm all about papers. I'm a neurobiologist, and I'm also really interested in molecular genetic techniques. And I need you to understand papers. I'm not here to try to teach you about classic neuroscience that you should have gotten all of that in your second year. I'm here to try to teach you what's going to be important in your lives down the road, whether you're in grad school or what you're going to encounter if you're going into science down the road. So we're going to talk about a few things. We're going to talk about these 3 papers. You don't need to memorize the titles nor the different authors. I am going to tell you a little bit about the authors just to give you some idea of how long they've been involved in it. You don't need to worry about their names. This is Chuck Gershbach, this is Amy Wagers, and then this is Eric Olson. I'm going to concentrate on Eric Olson's paper, mainly because I'm really fascinated by Eric Olson and his sort of approach to science. Sorry, you know, people who come in late, you actually have to eat, so you're going to have to grab stuff. Yes, yes, thank you. Um, so we're actually, some of you will have had this in HMB 300 with me previously, and you will recall Amy Wagers and her GDF 11 papers, and we will come back to some of the elements of her papers later on in the course. She has one of the papers that I just showed you on the previous slide, but Eric Olson at UT Southwestern has actually been involved in muscular dystrophy research for a number of years. It's not something new. He's been in this field for a very long period of time. He had a 2014 paper using CRISPR-Cas9 to use homology-directed recombination to actually repair in the germline, in embryos, before they become zygotes and then full-fledged animals, the fully functioning animals, repair. And it reason why, there are a number of reasons. Can you think of like 1 or 2 reasons why it's important to do all these papers? All 3 in science were done in the adult animal. Why in the adult? As I just told you, Eric Olson did this at the germline stage, so it's not that it's technically difficult to do. He's done it. He published 1 year before this paper came out. Yes, at the back. Absolutely. So the relevancy to clinical practice and definitely in individuals that are suffering from muscular dystrophy or Duchenne's muscular dystrophy or Becker's muscular dystrophy, it makes more sense to do it in the adult. And then there's the bioethical issue. Right now there is currently an embargo on doing any germline experimentation using CRISPR-Cas9. When that embargo will lift, or if there are individuals that are unethically doing this down the road, and what the consequences are going to be, we don't know. But right now, no one is allowed to work on the germline. So just again, as a recap, Duchenne's muscular dystrophy, the one that we started off with, and Becker's muscular dystrophy tends to be a fatal muscle disease. Becker's a little less so. It's X-linked, and it affects about 1 in 3,500 individuals, primarily boys that have that muscle wasting. Cardiomyopathy and heart failure are also common, as I mentioned to you earlier, and what this is a mutation that causes the premature termination of the dystrophin protein. And what that means is that because that linker molecule is no longer there and it's no longer able to cause the proper association of different molecules, that every time the muscle contracts, the muscle gets damaged, and it also ultimately leads to the loss of muscle membrane integrity and the degeneration of these muscle fibers, particularly by the time they reach teenage years. So this absence of this dystrophin causes this muscle degeneration that is so characteristic of Duchenne's muscular dystrophy. So a number of therapeutic approaches have actually been attempted in the past. And again, you're looking at this in 2016 thinking, well, you know, they've just done all this stuff. But the reality is, really, really smart scientists, including Eric Olson, have been thinking about this problem for a very, very long period of time. And yes, he published in 2014, but the sheer amount of research that has gone into this by a number of different labs, not just the 3 that I showed you here, people have been trying to do this for well over 2 decades. Like, once we understood what the mutation was, people tried to figure out ways around this particular protein, particular problem. One of it is the sheer size of the gene and therefore the sheer size of the dystrophin protein. It is enormous. It's one of the biggest genes. It's one of the biggest proteins. When individuals have done this in the past using like RNAi technology or antisense oligo technology in similar ways, you have to keep injecting an individual with those oligos. And that's not something that clinically we want to be engaged in. Now, if you recall from what I told you on Tuesday, when Roger Nickel and Feng Zhang were doing their experiments, they were able to see that the knockdown of the gene that they were looking at stayed knocked down for very, very long periods of time. So in theory, CRISPR-Cas9 stays in the system and lasts for long periods of time. And so individuals that theoretically get CRISPR-Cas9 therapy might not have to get injections multiple times because it's going to be lasting a long time. Now, again, I'm not going to ask you to go into a lot of detail. I'm going to tell you that this is probably a slide that you want to come back and review because some of you, it might have been a while since you've taken genetics, but we've already talked about at the very top about using this single guide RNAs to attack one strand of DNA, and that we know that the nuclease will actually cause a double strand break in a region, and that results in an indel if we want to eliminate the gene from being expressed. And those indels were important. Indels were shown by both Roger Nichols' group as well as Feng Zhang's group to occur after Cas9 directed double strand breakage. Now, one of you had asked at the end of the last lecture, so that's all about taking out a gene and putting it out of action. Can you actually replace a gene? And in fact, Eric Olson's group in 2014 in the germline, they actually used homology-directed repair. I'm going to use the abbreviation HDR, where you have regions that will homologously bind to the regions that you've excised. And in this particular case, because there's a template, that gene becomes repaired. However, in the cases that we're going to look at, we have no exogenous template. We do not have this homology-directed repair. And what happens is the gene simply just repairs itself through this non-homologous end joining. It just literally zips itself back up. So if we had something to replace it using homology-directed repair, we would be able to insert it into that broken region. But otherwise, if we don't have this template available, it will simply join back together using non-homologous end joining. That is an inherent property of the way in which DNA repairs itself. So again, Olson's group was the first in 2014 to use homology-directed repair in the germline of MDX mice. It worked fantastically well, but you can't do that right now, both ethically and in terms of human experimentation. We don't know if we can. We think we can, but in terms of being able to use homology-directed repair in humans, that hasn't been done. So in mice, it has. Technically, we think we can do it, and people have done it. And I've shown you Junju Huang. He has worked on germline human cells, but no one is allowed currently to do this for ethical regions. So germline genomic editing right now in 2016 is really not all that feasible. The other thing that makes this hard is this homology-directed repair is technically difficult in both heart and skeletal muscle tissue. And so other methods might have to be applied. So you can't necessarily go in, repair, because we won't go into all the details. The heart and skeletal muscle tissue don't really like using homology-directed repair methods. So in this particular paper that we're taking a look at, they're just using CRISPR-Cas9 to get this non-homology end joining, when they call it myo-editing. Literally, they're doing CRISPR-Cas9, taking out snippets of DNA and allowing it to self-anneal. And they're doing this in postnatal muscle tissue after delivering these viral components. And hopefully now, after a few lectures, this is becoming a little bit more for you. When I mention things like tropism, what does that mean for you? So this virus exhibits tropism for muscle tissue. What is tropism? Ingrid? So it's specific or selective for the muscle tissue. That's right, and it likes to infect muscle tissue. So these adenoviruses, and we've had this discussion several times. Why use adenoviruses? Well, we want to work with muscle. These adenoviruses tend to be specific for these muscle tissues. Lentiviruses don't really care about what tissues they're infecting, etc. So there are rationales and reasons for why these different elements are in these particular papers. Hello. Sorry, we have a long-standing tradition in this class. Okay. So, hi, we need you to take some of that. Yeah, that'll do. Thanks. Thanks. Okay, so this gets back to this idea. So dystrophin is large. In the past, we've had issues trying to repair large proteins. Can we actually use this to our advantage, though? Because it's so large, maybe dystrophin contains different regions that are either essential, we need it, we absolutely have to have this in dystrophin, and maybe there are non-essential regions. Maybe there are regions that if it's not there, maybe we can do without them. And we know, for example, that in muscular dystrophy, we have smaller versions of dystrophin around. So other studies, not in this paper and not in any of the 3 papers that I showed you earlier in Science, but other previous papers have suggested that if you could engage in exon skipping, where you are able to get the ribosome to get past a nonsense mutation, or to eliminate a stretch, that patients might actually benefit from having these smaller versions that don't have these essential regions, that don't have these non-essential regions around anymore in this particular context. And again, I don't need you to memorize this. This is be a really bad diagram for you to memorize. This is not a type of labeling diagram that I need you to go crazy trying to memorize. There are elements that I do want you to recall, but trying to memorize all of this stuff down here is not that important. We know that there are essential regions, particularly the ones on the N-terminal region of the protein, as well as on the carboxy terminal or the C-terminal region of the protein. We definitely need actin binding. That's one of the reasons we have dystrophin. It links this complex on the membrane to the actin filaments that are inside the myofibers. And then we have these different hinge regions, hinge region 1 as well as hinge region 2, and then we have these whole hosts of other domains in between, which are the rod domains. And we have multiple rod domains. And if you're thinking back to utrophin, we know that utrophin looks similar to this, but the rod domain itself is smaller. We don't have as many of these different rod domain units. So if we look at human and we look at mouse, the mouse, one of the things that you can see is, I'm hoping that you've all taken genetics and this is somewhat second nature to you when you see this, that if we're taking a look at human and mouse, there are these alternate splice regions where you need a certain directionality. There are acceptors and donors for splice regions, and we have other exons where it can be just like any other Lego block can bind to it. It doesn't matter what is next. So some things we need side by side, in other words. So exon 21 and 22, there is an acceptor and a donor that we need, and other exons, we don't care. It could be exon 23 and exon 24, 25, they all look identical. We could do without one of these different exons. And in the mouse, if you recall, it's really exon 23 that is at issue in the MDX mouse. In the MDX mouse, we have a mutation that occurred, and it causes a nonsense mutation here where we get premature termination of the protein. And so we don't get to form the full protein. We lose everything downstream of it. And so the idea is actually based on something that is really controversial. So I know some of you are in the GGB stream, right? Or the genetic stream. Have you heard of Adalorin in any of your classes? Adalorin, yes? Adalorin is a drug, and this Adalorin, it's really controversial right now. It is in clinical trials. It is not a It's not a gene, or it is not a group of nucleotides. It's actually a drug compound. And adelorin is thought somehow, magically, we don't understand the mechanism, to be able to allow ribosomes to go beyond or move through a nonsense mutation that has a premature stop codon built into it. So cystic fibrosis, for example, some forms of cystic fibrosis, they have a premature stop codon. They don't form the full transmembrane domain. Same thing in muscular dystrophy. There is that premature stop codon, but if you are given, instead of stopping here at this premature stop site, adelorin somehow causes the ribosomes to be able to push all the way through. And this was one of the first sort of allowing you to get this larger, more fully functioning protein. And this was the first idea. If we could skip by this particular blockage, if we could bypass it, it's like taking a detour around it. And as long as the road that leads back to where we should be going is the same, that we might be able to get the appropriate translation of the protein that we need. Now this gets into today's paper. And this gets into this idea of exon skipping. So rather than try to fix the problem, so all of you are thinking very linearly, like, wow, you know what we should do? We should create an indel, create this little gap, and then put in a full gene that will allow you to fix this premature stop codon. All of the papers that were published actually used this approach. They said, well, you know, if this is what the genetic sequence looks like, and some of these regions are ones that are non-essential, can we get away with taking out and skipping by that one exon that codes for one of these raw domains, and can we recover function without having to replace a defective gene? And so in this rationale, in exon 23, and this is sort of diagrammatically all of the exons, the 70-ish exons generating the dystrophin. There's an issue here in exon 23 where there's a premature stop codon. Can we simply use CRISPR and Cas9 to cut it? And if we cut it through non-homologous end joining, when the DNA anneals back with itself and we've skipped and bypassed this premature stop codon that no longer exists, it should start translating the rest of the protein. And can we act using CRISPR in the adult? And that's the basis of the paper. And it's to generate this internal CRISPR-Cas9 deletion through editing and then bypass, eliminate this particular exon. Is that okay for everyone? Do you recall the details from a week ago where we talked about the MDX mouse and having a premature stop codon in exon 23? Okay, so in theory, if you delete exon 23, you should be able to get everything back in frame, should be able to get dystrophin going again in these mice. Now, if you have read this paper, and it's optional, you don't need to read the paper. I'm going to cover what you need to know for your midterm. But if you've read the paper, I need you to understand a couple of things. This diagram up here actually shows that there are, okay, so you cut here in the exon, out here beyond the exon. And in fact, the guide RNAs, supplemental methods, is that you are generating, there is one to the premature stop codon, and that is the sgRNA MDX. And then there's another region out here, the R3 region, which allows for these two cuts. When you remove those two areas through CRISPR, everything lines back up through non-homologous end joining, and it's like bypassing or skipping this entire exon. And again, just for your information, so they're still using this same adenoviral system. They're using a truncated version of the cytomegalovirus promoter, which allows really high expression levels. It's still that pyogenes version of the Cas9 that we talked about. Inject this, and you also have to inject this. Just like Roger Nichols' paper, you need one vector containing the Cas9 nuclease, and then you need another vector containing the guide RNAs. And when they say sgRNA here, they actually mean sgRNA MDX plus the sgA3. Okay, and again, they tag this so that if the cells get infected, they will all glow green with this guide RNA. Okay for everyone? Yes, no, maybe? Maybe yes, maybe no? So they have some really cool protocols. And again, I'm going to make use of your knowledge of physiology, so we'll talk about this in a moment. So remember, they're doing this in the adult, and it is in the adult mouse. They generate these different viruses, two viruses or viri, so one containing the nuclease, and they also package another virus that contains the guide RNAs to excise exon 23. And then they do 3 different protocols, okay? And they're looking at the effectiveness of 3 different protocols. One is IP. Those of you who have done research, what is an IP injection? Anyone? I know you guys have done IP injections. Yes. So intraperitoneal into the abdomen or the abdominal space, in other words. There's another one that is done intramuscularly or IM, and this is the one that you would expect that it should work. And then there is this other one known as retroorbital. So tell me a little bit about retroorbital. You are injecting behind the eyeball. Why would you do that? It's technically very challenging. I've only done this, by the way, in a research setting once in my life, and I can tell you, even though I had a lot of other surgical skills, this is challenging to be able to do. Retroorbital, like you open up the eye and then you inject right behind the eye muscle. Why would you do that? What's the advantage? So you have IP injecting into the abdominal cavity, you have one being injected into the muscle, making sense. Remember, it's a muscle wasting disease. And then you have another one that's going retroorbitally. Why retroorbital? Yes, at the back. Does it reach the brain? Ah, interesting question. No, it doesn't, but we'll get to some proof of that at the end. It's a good thought. What do you know about the vasculature back there for those of you who are doing anatomy or neuroanatomy? Yes.
The blood vessels, they circle around the orbital, so where they don't cross over retina, they go around.
Okay, so they go around it, staying in the systemic circulation. So this is one way in which you can get systemic circulation. That's important. So they do these injections when the mice are at P1, so 1 day after they're born, then they go back and they examine for the expression 4 weeks later or 8 weeks later. For intramuscular, they will inject when the mice are 12 days old, and then they'll go back 3 weeks after they've injected and 6 weeks after they've injected. And for retroorbital, they will inject just over 2 weeks of age, and then they'll go back 4 weeks later, 8 weeks, and then 12 weeks later. So they want to see the expression of what happens after they do these injections. So again, I don't need you to memorize the end results of these particular result slides, but I need you to understand why they do these types of things and how we tell certain things from these slides. So at the top is immunofluorescence, although they call it immunohistology, and at the bottom in purple is this hematoxylin and eosin stain. So they take the muscle out, and then they want to see what happens when we do all these different things, in this particular case, 6 weeks after injection. So this is what wild type dystrophin looks like. Each of these, this lattice structure shows you where dystrophin is supposed to be on the outside part of the muscle. If you take an MDX mouse with no kind of manipulation, dystrophin is gone. We know that. And dystrophin staining is almost completely aberrant by this or absent at this age. But if we went in and we did injection into the tibial muscle, the tibial anterior muscle. And what we would see is after 6 weeks, this is the dystrophin that has come back. Okay, this is an MDX mouse now with exon skipping. We've removed that premature stop codon. It has repaired itself through non-homologous end joining. Dystrophin has been allowed to express itself, and this is the. Is it impressive or not? Imagine Eric Olson's in this room right now. It's pretty impressive, right? Eric, listen to this later. Students gave you a big thumbs up. So a couple of other things that we need to talk about. When you do H&E, you don't do this just to see what the structure of the myofibrils look like. It's not a very good stain for myofibrils. However, in histology, these H&E stains are actually really good to look at inflammatory cell invasion. It's good to see where the position of these myofibrin nuclei are located. And this actually tells us something, that without this intervention in the middle panel, what we see is that there are a lot of inflammatory cells coming in because it's degenerating. Remember, it's necrotic. And that we don't see that with the CRISPR-Cas9. And in addition, and it's hard to see on this particular diagram, a lot of the nuclei for the myofibers are misplaced. H&E staining tells us about inflammatory cells, and it tells us about cellular structure under normal conditions. So again, at about intramuscular 12 mice, if you take a look, and this is where you see the guide RNAs. The guide RNAs had GFP tagged to them. What this is really showing you is what the time dependency looks like. So if you look 3 weeks later, yes, you'll see dystrophin coming back. You see these sort of structures that you would normally want. But then after, and if you've left it and now go back in and stain at 6 weeks, you see that there is much more dystrophin. There is much more organization to this, and we start to see this really nice mosaic pattern down here by about 6 weeks. And it doesn't sound like it's really fantastic, right? So it's about 7%, and it might go all the way up to about a quarter, but it is still enough to cause a behavioral change. And I'm going to show you that in a few moments. So again, you need to be able to convince individuals that this is working, and certainly dystrophin is there, the staining is there, and it's not there in the MDX only mice. But if we were to take a look using PCR, and we were to go and take some of the muscle tissue that after injection, and if you examine it by RT-PCR, you will actually be able to see the correct size product using PCR. And you can see that it comes up, the longer you leave it, the better the expression is of this exon skipped. 23 is not there, and we can just around the 22 and 24 and see if we get a proper gene product. And you can see it gets much higher after 12 weeks. Then it starts off at 4, 8. You can see the slow rise in this particular through RT-PCR. If you then take some of those RT-PCR products, you cut the gel, and you take out the RT-PCR product, you send it for sequencing, it should tell you that the exon 23 isn't there. So one way in which we do PCR is to make sure that the size of the predicted PCR product is correct, and that's good. But because we might be looking at other artifacts, we want to make sure that what we intended to have is actually occurring. And so when they do this experiment, and they take some of these PCR products and then they do full sequencing, it is indeed exactly how they thought. Exon 23, which is a premature stop codon, is now completely gone, and the ends between 22 and 24 have completely annealed with each other, and we have almost like a continuous gene expression as a result of this. It's been spliced back in at the appropriate site. So again, these are long-lasting changes. They don't disappear with time. And the really cool thing is that they make note of in this paper, It doesn't matter how you do it. If you do it intraperitoneally, if you do it intramuscularly, which makes a lot of sense, right? Your muscles are wasting away. Let's do CRISPR-Cas9 on the muscle. Let's do exon skipping there. But if you also do this retro-orbitally behind the eye, you get just as good a result. Again, for me, it was really surprising, to be quite honest. So this is what the wild type normally looks like. The mice that don't have any issues, normal dystrophin, MDX, rapid degeneration. And by 8 weeks or 12 weeks, we see these necrotic autofluorescent markers that you see here. Let me ask you this. We see more of this autofluorescence necrosis going on here in this panel at 8 weeks than we do at 12 weeks. Why is that? What's going on? Yes, Arslan.
There may be nothing else to die.
Yeah, most of the cell death has already occurred. We've gone. We're sort of down at beyond sort of the maximal cell death that we can detect. There's not much more left to die. So if we do this retroorbital injection behind the eye, not into the muscle, and then we go into the muscle and now check for dystrophin expression, you can see that it is just as effective as injecting into the muscle. Systemic delivery through retroorbital injection is just as good, in other words, as directly into the muscle. Not only that, it also repairs the heart. They also check the heart. I'm not showing you those results, but the cardiomyocytes were also repaired using this retroorbital. Are you impressed by that? Pretty impressive, right? But again, you need to be beyond that. So if you're being a really good neurobiologist, or you're going beyond just the molecular biology and genetics, you also have to demonstrate that there is a functional change. Now, I'm actually going to interrogate you here beyond this one slide. One of the things that you see, you know, Jeff, the one thing I was thinking of was the number of stars that you see here during this particular test. And in fact, these asterisks tell you that the statistical significance is actually really, really good. This goes back to your earlier comment in the last lecture. But if we were to do this in either male or female, and again, I won't go into all of the rationales for why they decide to also look at female mice, but if you take a look, AAV, it doesn't bring it all the way up to wild type. So this is where they're measuring grip strength. Grip strength in mice is like finding out like how much muscle do you actually have? How much of your muscle is functional? So if they check grip strength measured in grams of force, so they allow the mouse to grab on to a bar that they can measure its grip force, the wild type would be way up here. The MDX, a similar age-matched MDX, is all the way down here, but we can see that after CRISPR delivery and exon skipping, that it's not bad. It's not perfect, but it's not bad. It's not as good as it should be up here, but it is significantly better than not having any of the dystrophin around at all. So it shows you that there is a behavioral consequence. Yes, their muscles actually do get stronger. It's not as good as the wild type. Maybe one day we will go from about 25% of the myofibers being rescued to 100%, and maybe we'll get all the way back up there. But right now, it's not bad at all. It's significantly better than it was with just the wild type. Sorry, with just the MDX mutant only. Now, this is one way to measure the effectiveness. How else could they have measured it? Think about this as a multiple choice test question. You should be able to, based on what I told you in the last lecture on MDX, to give me an answer. How else do we measure for muscle loss? This is a behavioral test, grip. What else can we measure? Doesn't have to be behavioral. Yes.
It's also looking for
The creatine kinase, we could look for that. And if we measure and see a decrease in the level of creatine kinase, that's great. And that's exactly the answer I was looking for. I didn't show you this data, but in other papers, the other two papers, they actually measured for the release of creatine kinase, the isoform, and you could do that. And I need you to think about this holistically. Just because I presented you with this data doesn't mean I might not ask you, how else could they have done this, right? And I want you to start thinking like scientists. Now, your colleague at the back asked this brilliant question. So does it get everywhere? And that's the question that they were asked. This paper, by the way, was published at the very end of 2015. If you go back and look when they submitted this paper, though, it was at the very beginning of 2015. It took them a whole year to be able to address the issues that their reviewers had 13 pages of supplemental figures. And some of this is, if you do these types of injections, intramuscular, retroorbital, or intraperitoneal injections, all of them work on different organs. All of them work on muscle. Doesn't matter where you're injecting. You don't have to inject directly into the muscle to do this. And it might be because they're going outside of the regular vascular circulation because of the AAV. And again, even in smooth muscle cells, we're able to see a recovery of function. But can we actually get this in the brain? So the first question is, do you have dystrophin in the brain? You do. So this is a wild type mouse. In red, you see the staining for dystrophin. All you're doing is looking, do you see dystrophin in a wild type mouse? And yes, you do. This is just normal staining. Dystrophin exists in areas like the hippocampus. It exists in neurons throughout different regions of the cortex, but particularly in CA1 and CA2 hippocampi, we see lots of dystrophin. In the MDX mouse, that dystrophin drops. There is a behavioral cognitive loss following dystrophin loss. But did that AAV, the IP injection that works so well in all of the previous diagrams I showed you, not complete recovery, but a lot of recovery, it doesn't work here. It does not cross the blood-brain barrier. It is excluded from the blood-brain barrier. So this has a lot of potential, but some of the cognitive effects that we know accompany muscular dystrophy might have to involve other routes. Yes, Danny. Oh, sorry, yes.
Instead of doing an oral injection, couldn't they just do an intravenous injection?
They could have. So actually, other papers use tail vein injections. Um, also technically difficult. If you've ever tried injecting in the tail vein of a mouse, I don't think you do that in any of your labs because small animal surgery labs in physiology, we were required to do that in our third year, but it was so difficult that they eventually dropped those labs. It's really hard to find. Do you have a question or a comment?
Yeah. Did you say that AAV was like specific to muscle tissue?
This particular isoform AAV9.
Yeah.
It's tropic for muscle. Other AAVs are tropic for other types of tissues. So smooth muscle, yes. Cardiac muscle, yes, and skeletal muscle, yes.
Yeah. So I was just wondering about the results about the brain. Even if it
Yeah.
could cross the blood-brain barrier,
Yep.
wouldn't.
There is a little bit of tropism for AAV in general, whether it's the 9 subtype or other subtypes for neurons. Neurons happen to be highly susceptible to viruses, and we'll learn a little bit more about that later on. So if you want to transduce neurons, viruses are a very effective way to be able to do that. Sorry, there are lots of questions. I don't know if I have time right now to do this because I have like 2 more slides and I think we have to leave at 1. If you can email me your questions, or I'll stick around at the end if you want to chat about this. So what does this all mean? So this means that this myo-editing using CRISPR-Cas9 works in adult mice. It's postnatal. And we get this persistent expression of this gene, the gene editing components. We see it getting better with time, not disappearing over time. This exon skipping by that non-homologous end joining was really good. And in fact, not shown in this paper, but with the companion paper that Eric Olson had in 2014, he actually says that this is better than when he used HDR, so homology-directed repair, when he put in a good copy of the exon 23. This actually was more effective at recovery than using HDR, at about 10-fold more efficient than what he saw in his earlier paper. That's pretty remarkable for him to be able to say that, because his Science 2014 paper was actually one of the most cited papers for using germline editing to cause recovery in the adult animal that had muscular dystrophy. So this myoediting seems to be really successful. So you'll have to think, like, what kind of disease conditions might we be able to tackle? What are some of the technological hurdles that are still remaining? So I want you to think a little bit based on what you've heard here today. Um, Today, I'm going to just mention from today's lecture, you are less likely to get a lot of multiple choice test questions. I'll probably ask you like 2 at most. It will be related to technical issues. Why might you, or you've seen this in a mouse and you get this kind of phenotype, what might you have used to detect inflammatory cells? All of you should right now be thinking hematoxylin and eosin staining, and that's great because that's what I want you to be thinking about, the technical issues, not the actual results from today. I want you to remember HDR, non-homologous end joining, and exon skipping, and I want you to be thinking about that as a short answer question from today as well. We do have a couple of minutes, so if you do have questions, I'll take them. And the other thing I'm going to ask you to do, because I do really want you to start thinking about your midterm. I'm going to be putting online this weekend the same types of things. One, like a checklist from this week's lectures. If you study that checklist and you know it really well, that checklist is enough for you to get 100%. Number 2, I need you on a separate discussion thread to talk to your classmates and then to figure out which groups you're going to be working in for your presentation, your group assignment, as well as for your post-midterm group discussion. The one thing I'm going to mention about that post-midterm thing, I'm expecting all of you to do well, but this is one thing you can't do. You can't take apart the midterm and say, you do the multiple choice, I'm going to do the short answer section 1, and I'm going to do short answer section 2. You have to be working on it all together, okay? And I need you to post the members of your group. If you don't have anyone, I need you to indicate that to me as well, because I want you to be working in a group. Okay, so we have like 2 minutes. Any questions that you have about the material, about your assignments, or anything else? Yes.
Since dystrophin also interacts with that big blob that we were talking about first lecture
Okay.
and the sarcolemmal membrane,
Yep.
can you also use that as a target to figure out if the wild type has coverage?
Could you use the other interacting molecules in the right place? And in fact, that's a great question. And I'm skipping all the way back, but in fact, this is what it actually shows. The blue staining there actually shows some of the interacting molecules, and yes, you could do that as well. Yep. Yes.
Will we be receiving a proper prompt to the group work, or are we really supposed to just get them to do it?
Sorry, I couldn't hear you.
Are we going to receive a proper prompt for the group work essentially, or are we
You will get instructions for every assignment ahead of time. Yes, you'll get some prompts and everything else. Yeah. Any other questions? Yes.
For short answer, is it okay to use abbreviations?
Yes, you can. If I use abbreviations, as long as they're consistent with the abbreviations we've used in class, you may. Okay, great.